Yeah, so I'm going to tell you about my practice that I like to sum up as making biohybrid machines or living computers. I think the Schrödinger definition that Chris showed is one of the most interesting definitions of biology, and it's basically about entropy, right? But entropy is a measure of structure in matter, but it's also a measure of structure in information. And I think this is the central point in all of my work, or at least where this thing started. So if biology is a system that is capable of organizing entropy, or like reducing entropy, organizing stuff, matter, it's also capable of organizing information. And basically that's one of the basic definition of what a computer is. So in that sense, you can look at biology as, or any living system, a lot of also non-living system, as computing. And so I'm going to show you different projects I did. Some of them are more engineering, others are more design and art, and some are in between, let's say, that all try to make this kind of living computers. And because, yeah, also something I didn't say is that I studied here, and I did some of these projects in the research lab that used to be at the 3rd floor and not here anymore. And so I'm going to start with this project that I did with Ariel Lindner, who founded this place. And yeah, so basically what we're trying to do is to make bioelectronic interfaces for bacteria. The reason why we wanted to do this is because, well, I come from interaction design, and so it's this discipline that make interactive installations. And most of the time, those installations involve sensors, electronics, softwares, but we don't really have ways to do this in an easy and controllable manner with biology. So for me, the most convenient thing would be to have an interface that allow me to plug biology to a normal Arduino, a normal electronic system. But they didn't exist, so we needed to make them, basically. So this is a schematic representation of E. coli. It's a bacteria. I don't know, yeah, we can see the mouse. So this is the membrane, right? There are pores in this membrane. Inside the cell, you have charged particles, and outside the cells, you also have charged particles. But there's a disbalance between those charges, which create a current, right? A potential energy. Ions wants to flow inside. And so basically, it means that every cell, bacteria, but every cells behave like a battery. And that's the voltage of the canonical voltage of E. coli. It's minus 150 millivolts. And this battery is used to do a lot of things in the cell, like from processing nutrients, growing, dividing, moving, like using flagellar motors, for example. So if you break this battery, a lot of things are going to change in the cell. And so this was the starting assumption of this research project. We built this device that is basically an array of 96 electrodes. And we can use that to send electroshocks to 96 wells of a 96 well plate and then analyze what happened to the cells, right? So we ran this experiment. We basically screened, I think, 8,000 different conditions. We had this library that was made 20 years ago that basically involved fluorescence reporter. You can check what genes is activated using fluorescent scan, like reporters, basically, upon electrical signalings. And so we found some. And then the idea around that is that we could have bacteria growing in microfluidic device. So you would have like the bacteria here. You overlay this onto electrodes, and then you can send electrical signals as an input to the bacteria, rewire what the bacteria is doing using normal tools of synthetic biology. So synthetic biology is the discipline that engineered biology for at least the past 30 years, making a lot of different toolkits to make computers out of gene circuits, right? So you can have an input and decide on what to do given this input based on digital logic that was originally made for computers. So for example, a simple example is an AND gate. It's just a gate that says if I have 2 positive inputs, then the output is positive. If I have just 1 positive input and the other one is negative, then the output is also negative. It's just basic computing paradigm. But basically, we could reproduce that using molecular biology with electrical input and then report that in terms of an electrical output too. And then because you have electrical in and electrical out and computation in the middle, you can make circuits. And there the idea was that, okay, so if we can have some logic, we can maybe at some point try to replace, you know, Arduino boards or any kind of electronics that we're used to use in our interaction design systems by biology. And the specific thing about biology is like it's not just about replacing old components, it's also capable of doing new things. So biology, most of the time, compute on the chemical realm. So it takes chemical signals as inputs, process them using chemicals, and output other chemical signals, right? Which is something that most of our electronics cannot do. So it's also trying to expand the toolkit of what the electronics can do, right? Okay, so I'm going to talk about a more artistic project here. I had the studio for living computation for about 4 years. That was mostly trying to work in the realm of speculative design, trying to make living computers for the sake of an argument. So it's not going to be something that you have to judge in terms of is it efficient, is it working? It's more a question of narrative and what if we could make these things. So I made this 5-story tower that is at each level comprised of an array of photosynthetic algae. These are called marimos. They are originally from cold lakes in Japan and Northern Europe. And so they are photosynthetic, and like anything that is photosynthetic, they produce oxygen when they are exposed to light. And this is what you can see on this picture. Here is like tiny bubbles of oxygen. But because of the structure of the algae sphere, they are filamentous, those bubbles of oxygen get trapped inside the sphere. And when there's enough of them, given enough light as an input, they start to float. And so this is what you're seeing here. And after a while, they sink because the bubble just burst. Time-wise, we're looking at like to start floating, basically 30 minutes of light exposure, and then you wait an hour, and then it sinks. So it has two states. It can either be like floating or sinking. And like everything that has two states, you can abstract to digital information, basically. And so here the idea is that using 6 of these algae balls and encoding zeros and 1 in the fact that they are either floating or sinking, you can treat them as a byte and then write a one letter using those states. So then I write them in 6 bytes arrays, and I build the entire framework around it. So at the center, you have a sensor array. On the outside, you have kinetic arms that can move around and up and down. Here are some lights, and so basically just addressing every bits of information inside the tower. Yeah, so altogether it looks like this, and so it's not going to replace a normal hard drive, but it is a hard drive, and so it works exactly the same way. It abstracts data into zeros and ones, but instead of storing information into transistor, it's storing information into living materials. And so to the best of my knowledge, I think it's also still the case, but it was one of probably, I don't know any other project that store information into living substrates. So like what people do normally is that they store information in DNA. Everybody heard of this thing, but DNA is not alive. And there's a good reason for that, is because it's not changing through time. And so when you store data, the data is supposed to stay the same kind of for thousands of years. And that's the interest. That's why people want to do this. In this case, it's much more complicated because when you write data, because of the dynamical nature of life, it will be erased through time. And so it makes you think about like the carrying capacity of your media, in this case algae, how much data you can write, how fast does it degrade, what does it mean for information storage technology, and in general, our digital culture, let's say. Okay, and so one last project I'm going to present is a project I did in industry at OXMAN in New York. So OXMAN is a lab, design center lab that was founded by Neri Oxman, who used to run this group at MIT, was called Mediated Matter. One of the first group trying to use synthetic biology in biodesign. And then this company spun off of this lab. And we were interested in volatile organic compounds. So volatile organic compounds are small molecules that are produced by almost every organism on the planet. They are just small and they are volatile, and so basically they pass through the air. Some of them are just byproducts of metabolism, like for example the smell of a rose, that also have like some signaling property, but and some other really have like some communication purpose. So here, the example I'm going to show is the one of green leaf volatiles. So everybody's kind of familiar with the smell when you cut the grass in front of your house. It smells something special. It's actually a cry for help that plants emit when they are under mechanical stress. And the reason why they do that is to warn their neighbors that a mechanical stress is coming. And then their neighbors can divest from growth and reinvest into mechanical protection. So the mechanical protection they evolve for is bugs. So they cannot do anything against you when you when you like, try to cut the grass. But yeah, so basically they really change their expression pattern depending on this, and this information traveled fast, traveled long, and this is why they do it. And so this experiment was really telling about it. So the researchers here put some green plants in a bottle here. They crush the plants, and then they push the air from one side of the bottle to a petri dish where Arabidopsis was growing. So Arabidopsis is a model plant organism. They engineered this plant with a calcium sensor. And so calcium is an ion that is often used to report on electrical activity. For example, in neuroscience, all of the images that we have of the brain, like a lot of them come from calcium signaling. And so here they engineer the plant so that it becomes fluorescent upon electrical signals. And so this video I'm going to show is basically they push the air and then the plant is exposed to those volatile organic compounds, and then we can image change in electrical behavior. And you can see that basically the signal is propagating through space at a speed that is pretty fast. And this is what's happening in every backyard when you cut your grass. And so here you literally see information moving through space, and what you don't see is what's happening inside the plant at the gene expression level. And basically, this is what I was telling you earlier, they will change their gene expression pattern so that they will invest into resistance instead of growth. So plants communicate using VOC. Those VOC carry some computational capability basically inside the plant, so we can make a computer out of it. So this is what we're trying to do. We build those devices that are bioreactors growing E. coli, so the same bacteria I was talking about earlier. But we engineered the E. coli. We took genes from plants that code for enzymes that produce those VOCs. And then suddenly our bacteria could produce the same smell as those plants. And what you see here is this is the liquid phase, right, where the bacteria are growing. And here is the headspace where all of these VOCs are trapped. And then we build this device here on the right. It's basically a mechanical lung that can suck up the air from one bioreactor and move it to another. And we also engineered E. coli to be able to sense smells from plants. And so when you stack all of them together, then you can basically build prototypical networks where you have bacteria that produce VOC, bacteria that sense VOC, and a mechanical device that move VOCs from one plant to the other. So that's the entire setup. And yeah, basically this is what we're trying to do, is trying to propagate the signals from one side to the other, try to build logic gate as well. So basically things that say, if I receive one smell and not another smell, then I do produce something else. And then you can start to do some simple mathematics. And the idea is that at some point, maybe we'll be able to do this at the scale of a forest. So you would have smell sensors that take the shape of a plant or a population of bacteria that you put somewhere in forest or different places, that process information from trees or from humans or from animals using only smells and report using other smells. And yeah, that's it.